Dear student, now we will see how the digestion get completed in a small intestine, and we will also discuss what is the role of pancreas. So here we will start with the role of pancreas. Pancreas is a heterocrine gland and it act as exocrine as well as endocrine gland. It consists of acinar cells and islets of Langerhans. Acinar cells secrete pancreatic juice. The secretion is stimulated when food entered duodenum. The endocrine cells of intestine secrete secretin and cholecystokinin, which stimulate the secretion of pancreatic juice. The islets of Langerhans secreted hormones like the alpha cells secreted glucagon, beta cells insulin, and delta cells somatostatin. Now, the digestion of food in intestine, if you see when the food enters the intestine, when the food combined with the gastric juice, it become chyme. After the digestion, the term we use for the partial digestion of the food and by the gastric juices. So chyme we use, which is acidic in nature, and this chyme enters the duodenum wall. The duodenum wall secrete two hormones called cystokinin, which stimulate the secretion of pancreatic juice as well as expulsion of bile from gallbladder, and duodenal gland gets stimulated and secrete the enzyme. So two things happen simultaneously here. The pancreatic juice is rich in bicarbonate ion and that help to neutralize the acidic effect of chyme, or you can see gastric juices. Okay, so duodenum has the secretion from the liver and pancreas, and it has pancreatic juice and intestinal juice. So first let's see pancreatic juice action. Pancreatic juice consists of proteolytic, amylolytic, lipolytic and nucleolytic enzyme, and the same enzyme, proteolytic, amylolytic, lipolytic, all these enzymes are also present in the intestinal juice, but the enzymes are different. Okay, so if you see, their action is different. So first we'll talk about the proteolytic enzymes. Again, I told you that they are mainly the proteolytic enzymes are secreted in the zymogen form. So here the two different categories of enzymes are there. They are known as endo and exopeptidase. Usually what is the difference between two? The endopeptidase, it hydrolyzes the peptide bond within the main peptide chain, whereas the exopeptidase, it hydrolyzes the protein either from C or N terminal. So slowly keep releasing either dipeptide or single amino acid from the terminal ends of the protein. Okay, so the enzymes, endopeptidases enzymes are trypsinogen and chymotrypsinogens, which are secreted in the zymogen state, and exopeptidases are procarboxypeptidase. Now let's talk about the proteolytic enzymes which are part of intestinal juice. So here enteropeptidase is secreted, which is not secreted in the zymogen state, enterokinase, which is also known as enterokinase, and proaminopeptidase, which is secreted in the zymogen form, and dipeptidase. These are the enzymes of intestinal juice. Now if you look at the sequence how these get activated, enterokinase or enteropeptidase, it now in the intestine all the juices are mixed. So enteropeptidase of intestinal juice act on the trypsinogen of the pancreatic juice and convert the trypsinogen into the trypsin. Okay, now trypsin act on the chymotrypsinogen, okay, which is the inactive form, and convert the chymotrypsinogen to chymotrypsin. The chymotrypsin act on the protein and convert the protein to polypeptide. Now on this polypeptide, the aminopeptidase, carboxypeptidase and dipeptidase acts and convert these polypeptide to either dipeptide or amino acid, which are the end product of digestion. Okay, so the difference between carboxypeptidase and aminopeptidase is that one, the carboxypeptidase is secreted by the pancreatic, whereas the amino by the intestinal juice, and aminopeptidase act at the N terminal, where the carboxypeptidase act at C terminal of the polypeptide or protein. So this is how the protein get converted into amino acid, which is the, now you can see here the one enzyme get activated, it activate the another. So this mechanism is called cascade of activation. Okay, now we will see what will happen to the carbohydratase action or amylolytic enzyme. So already partial digestion has already taken place. So we have the carbohydrate in the form of either amylose, amylopectin, or maltose, or some other disaccharide. So amylose, amylopectin acted upon by the amylase or an isomaltase and convert it into the disaccharide and dextrin. So further disaccharides are acted upon by separate enzyme maltase, lactase, sucrose, and they convert them into the respective monosaccharide, and these monosaccharide are the end product of digestion. So this is how the digestion get completed in the small intestine of carbohydrate. If you see the lipid digestion, already emulsified fat is there, which is acted upon by the lipase, because this conversion has already taken by the bile, which is present in the intestinal juice. Now lipase from both pancreatic and intestinal juice will act and convert it into the fatty acid and glycerol, which are the end product of digestion. Similarly, nucleosidase and nucleotidase are there. The DNAs and RNAs act on respectively on DNA and RNA and convert them to the respective nucleotides, and then nucleotidases act on them and convert them into the nitrogenous base, sugar, and phosphate, which are now the end product of digestion. So here the digestion of each and every component is completed. Okay, so now after the digestion the absorption occurs. So we'll be talking next about the absorption of food. Thank you.